Let's start our conference with the name of Allah.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان والأرض وضعها للأنام فيها فاكهة والنخل ذات الأكمام والحب ذو العصف والريحان.
Subhanallah. Now let me introduce you our organizers.
Assalamu alaikum and good morning, everyone. It is my pleasure and honor to invite you at first International Conference for Genomics of Beneficial Microbes hosted by NIGAB NARC. NIGAB National Institute for Genomics and Advanced Biotechnology is an exclusive national institute dedicated to undertake agriculture research in all the three domains of life, including plants, animals, and microbes. It is a complex of biotechnology with 28 state-of-the-art labs, each designated for a specified area, along with glasshouses and containment facilities. Since its inception in 2007, NIGAB has been recognized nationally as well as internationally for quality research in agriculture biotechnology. Research programs for NIGAB primarily focus at enhancement plants and animal productivity by 10% at least, and value-added bioproducts for national food security, thereby helping in achieving sustainable development goals for better economy in this country. Through traditional food genetic engineering, Microbes has developed GMOs of wheat, groundnut, potato, and tomato against disease, drought, and salinity, which await approval from regulatory body for commercial utilization at farm level. It has also exploited marker-assisted selection for development of new varieties of crops for rest, salinity, and drought tolerance. Microbes has embarked on new breeding technologies, including speed cloning, speed breeding, genomic selection, and genomic editing using CRISP, PR, Cas9, BCas9, CPF1 approaches for enhancement of plant productivity. Next generation sequencing data obtained through genome sequencing and transcriptome sequencing is being analyzed for varietal hybrid identification, SNP, marker development, and gene discovery. 30 novel genes related to various desirable traits in plant have been isolated and being genome edited in plants. NIGAB is a credible reference laboratory of biotechnology and molecular-based diagnostic testing of GMOs, identification of unknown olive varieties, and adulteration in exportable quantities for quality assurance. Tissue culture section has replaced infected banana with virus-free banana in Sindh province, and currently 4 new varieties of banana are in pipeline. In livestock sector, NIGAB is actively involved in developing vaccine for disease prevention and epidemic control, and also in selecting superior breeds of animal using NGS data for improved milk and meat.
I would like to invite Dr. Shakra Gazanfar for welcome speech. Dr. Shakra is Senior Scientific Officer at National Institute of Genomics and Advanced Biotechnology. Her area of research is development of target-based probiotics using next-generation sequencing. She is one of the pioneers who have initiated animal probiotic research to improve livestock productivity in Pakistan. Over to you, Dr. Shakra.
Okay, thank you so much. Assalamu alaikum and good morning to everyone. Okay. Thank you to each and everyone for being here with us. And today is our first international conference on genomics and of beneficial microbes. We are proud to be able to host it here at NIGAB NARC Islamabad. Okay. Our goal is to bring together different types of scientists from all over the world to present and exchange the valuable information regarding our related to the beneficial microbes and their genomic analysis. So the second aim of the conference is to study the safety analysis of microbes based on whole genome sequencing. Secondly, the microbial gene sequencing data analysis with the help of available, you can say, software, and then gathering of the scientists, students all over the world who are working on the beneficial microbial genomics. So these are the keynote speakers and presenter of our today's conference. Is a special thanks to the chairman PRC, Dr. Ghulam Muhammad Ali, for his interest on the conduction of one-day conference on the genomics of beneficial microbes. Thank you so much, Dr. Saab. Now, what are beneficial microbes? Beneficial microbes are, you can say, the probiotics are the live microbial feed supplements. And these are among the fast growing health food based on the use of beneficial microbes. The global market for beneficial microbes, or you can say the use of beneficial microbes for the production of probiotics based product is growing rapidly, and you know that it's estimated to reach into $3.5 billion by 2030. So, but the basic reason behind the probiotic use is the safety issues. Number one, safety issues of probiotic product has been reported frequently in many years related to Pakistan and other things. Secondly, the inadequate taxonomical identification, or you can say the wrong identification of the probiotic strains leads to the very poor production in the probiotic product. No. Third step is the probiotic genomes variation can lead to and then would affect the probiotic function and in terms of its stability and its viability. Second, antimicrobial resistance genes and resistance factors are the used to human, which leads to the muting in the screening of probiotic product. No. The agriculture-based product containing virulence factor can lead to the pathogenic transfer from farm animals to the human. So what is the importance of microbial genome? Whole genome analysis is a useful and wonderful method to study and to screen and to evaluate the quality and the safety of any type of probiotic product, whether it's a human-based probiotic product, animal-based probiotic product. So we should use whole genome sequencing analysis to analyze the, you can say analyze the safety of the probiotic product. So this slide show the whole genome sequencing determination of the safety determination of the bacterial strain. From that figures, we can easily identify that the beneficial bacterial strain should be screened out for different types of genome, like the beneficial genes, like the pathogenic gene. From nucleotide sequence to potential gene, we must use the whole genome sequencing, and from whole genome sequencing, you can easily identify whether your bacterial strain is safe for the preparation of product or not. And then from nucleotide sequence to potential gene, and from open rate frame to amino acid sequence, you should be study the genomic analysis. And from amino acid sequence to protein function, and from these data, you can easily interpret the result with the help of the whole genome sequencing. And from that, you can easily identify that whether your bacteria is safe for production of any type of product or not. So from a single portion of a bacterial genome, you can easily identify the gene related to the probiotic potential and gene related to the pathogenic potential. So from that whole genome sequencing analysis of any type of bacterial strain, you can also identify the cellular function based on annotative genome sequence. So you have to study the annotation of the bacterial genome, which will you use for the probiotic production. So this slide will show that we should use different types of method to study the safety analysis of any type of microbial gene strain. Number one step is the identification. You should identify your bacterial strain based upon WHO requirement. You should identify with the help of whole genome sequencing analysis. So secondly, the probiotic gene variation and other types of things should be identified with the help of whole genome sequencing analysis. From that data, you can also identify the probiotic related gene. And then from probiotic related gene, you have to find out whether any type of antimicrobial resistance genes are present in our microbial strains or not. You have to identify. So after that, these steps, you can use that bacterial strain with the help of whole genome sequencing analysis identification for preparation of any type of product. So this slide shows the brief introduction related to the phenotypic identification, molecular identification, probiotic activity of the bacterial strains, the whole genome sequencing analysis of the bacterial strain. And that, of course, from that, you can easily prepare that bacteria for development of any type of probiotic product, which can be used for feed supplement or food supplement. So the brief of the whole genomic sequencing, you have to study with the help of Galaxy software server. It's easily available online and it's totally free. Genomic assembly with the help of Galaxy server, then the genome and assembly with the help of different types of online available tools. And then bacterial genome annotation with the help of a ROT server, it's also free. Then determination of selective virulence gene, determination of main virulence gene, and of course, after that, you can easily analyze that my bacterial strain or you can use yeast strain is very much capable to develop any type of probiotic. So more comprehensive talk will be start in half an hour. And then, I will introduce the beneficial bacterial genomic initiative, like we will plan to online activity on beneficial microbial genome, like online seminars, talks, special event related to the beneficial microbial genome, and then at the end, and hands-on training on the microbial identification based on next generation sequencing will be done. And after that, each certificate will be provided to each participant. For any question, suggestion, and clarification, students, researchers, please feel free to send us email at microbial_genome_nigab@yahoo.com. So once again, a very warm welcome to each and everyone for you. If you have any question, please feel free to email us. Thank you so much.